The aim of this procedure is to purify recombinant His-tagged proteins in a high-throughput manner using an automated protocol on a robotic platform. This is accomplished by first overexpressing the proteins in E. coli using small volume cultures. Next, the cells are chemically lysed to release the proteins that then bind to magnetic nickel beads. Then, the beads are washed to remove contaminating proteins and to enhance the purity of the desired proteins. Finally, the proteins are purified from the magnetic beads. Ultimately, results can be obtained that show the concentration and purity of the proteins through BCA assays and SDS-PAGE, and additional functional assays to determine the activity of the proteins.
The main advantage of this technique is that we can now purify a large number of proteins under identical conditions with minimum intervention. This allows us to obtain a high degree of reproducibility, and it also minimizes human error.
Well, this method is particularly useful to study large numbers of point mutants of a particular protein, as it ensures that variability in the activity of the protein is actually due to the mutation and not to differences in the preparation of the samples.
To prepare overnight bacterial cultures, begin by filling 24 well plates with water and microwaving them for 2 minutes to sterilize them. Afterwards, discard the water and microwave the plates again for 2 minutes. Using freshly grown lac-inducible BL21 Rosetta 2 bacterial colonies or frozen glycerol stocks, inoculate 1.5 milliliters of auto-induction medium containing a mixture of glucose